So I covered a lot of this at the end in the overview of gastric digestion. But this video begins with taking this apart and looking at each one. So what I've done here again is just kind of piece by piece, is know that the cephalic, which is really controlled by the head and the brain, is going to occur before food enters the stomach. And like salivation, the smell, the taste, the thought of food will start this process from the medulla. So the medulla is directing everything that's going on here. And you will begin to stimulate the gastric glands through the parasympathetic nerves, right, vagus nerve especially. And you'll start the release of mucus, hydrochloric acid, pepsinogen, intrinsic factor, and gastrin in the stomach. All right. So this is a very short. If you don't eat, the cephalic phase will end, and we won't have any of the rest of the phases because they're obviously pointless unless you eat something. All right. So the second phase is, again, food enters stomach. This is going to go on for several hours, whereas the cephalic is not very long-lived at all. So now, stimuli again for this gastric phase is distension of the stomach. So food is in the stomach, liquids are in the stomach. Partially digested proteins, because again, proteins are basically what are targeted by the activity of the stomach, and a rising pH. So every time you eat, the pH of the stomach is going up. And when the pH rises, that actually causes the stomach to produce more hydrochloric acid. And because, again, the digestive process, the chemical digestive process, has to occur at a low pH in the stomach. So here again, stimuli stretch the stomach, food and rising pH will activate the different cells and reflexes, and you'll have increased secretion and mobility. It said gastrin is directing most of that, which targets the parietal cells to keep on making hydrochloric acid and bring that pH down. Right? Smooth muscle is stimulated, and if, of course, the pH, I mentioned here, pH drops too low, right, excessive acidity, then gastrin secretion stops, and then you stop making hydrochloric acid. The intestinal phase, I call this self-preservation, right, for really the intestines. So the intestines are in charge. This begins when the intestines are stretched, when there's acid present in there, so get hydrogen ions. Fats, partially digested proteins, all can trigger this as well. So we're looking at this right here, okay? Because this is really going to be an inhibitory, so inhibit stomach secretory activity in the intestinal phase. So we call this the gastroenteric reflex because the small intestine is telling the stomach, slow down, we're not ready, right? And so now you'll see sympathetic impulses are going to be prominent, all right? And these will tighten the pyloric sphincter during this phase, and the intestine will start releasing secretin, cholecystokinin, gastric inhibitory peptide, right, to inhibit any more gastric secretion and movement. And this, again, will be done so that we can protect the small intestine from the contents of the large intestine, contents of the stomach. All right, so digestive function overall here, the rate of emptying depends on the type of food and fluidity. It's generally 2 to 4 hours from the time we enter. I said fats take the longest, lipids, which are fats and oils, take the longest to leave because digestion is slow in the small intestine, and so they are slowed down. When you have a lot of fat, that slows down, that really activates that intestinal phase and slows down the emptying. Mechanical digestion in the stomach, a mixing or churning is what we see happening, and that liquefies. And so as the process begins in the stomach, the two sphincters, the gastroesophageal and the pyloric sphincter, tighten, and we just see this repeated waves go back and forth that just pummel the food into, again, a semi-solid to a liquid. Gastric emptying, so moving it from the stomach to the small intestine, you see just a little bit of chyme entering the chyme. And the chemical signals and stretch there initiate that enterogastric reflex, and that will inhibit some of the secretion. So as the chyme is squirted through, the small intestine begins to take control of what's going to happen next and how quickly, right, food is going to move from the stomach to the small intestine. Chemical digestion in the stomach is, right, it's really, I say, we sit for a little while before mixing, so that salivary amylase that was effective for carbohydrates functions a little bit longer. But once the chyme becomes acidic, right, we get enough of the parietal cells making hydrochloric acid, amylase is destroyed and lingual lipase will begin. So again, you had secreted this in the mouth, but it wasn't working yet. And it has very little activity. It's really what we call the short chain fatty acids here. Most of the activity, chemical activity, is through pepsin that works on proteins, an intrinsic factor that helps us with B12. All right, so we're going to skip the cahoots and move into the small intestine, right? So this is really considered our primary digestive organ because it's really, we haven't done a whole lot chemically when we're nowhere near ready for absorption. So this does a lot of the job. Now they've color-coded this for the 3 regions of this very long tube. And so duodenum is the shortest and it's the first, connecting us to the stomach. And it has a number of modifications for its function, including lots and lots of these mucus glands and lots of ducts from the liver through the bile duct and from the pancreas. The jejunum is not quite as long as the ileum, but again, here's where we do a lot of absorption. It's between the duodenum and ileum. And then the ileum has lots and lots of blood vessels for absorption, lymph nodes to help for protection, and this will join the large intestine at an ileocecal sphincter, which is between the ileum and the cecum, the first part of the large intestine. Mesenteries are these organs that are really between all these and around all these, this tubular structure. They are technically 2 serous membranes back to back, but they produce a lot of different cells that help direct the functions of the organs of digestion and other organs of the body. All right, so modifications to our wall, especially the mucosa and some of the submucosa. So here's, right, fairly low magnification, higher. So here's a villi here, now they're enlarged here. These are the, right, microvilli that would be on top of one cell here, right? Showing here's a cartoon, here's where they look like. That's these guys up here. All right, so, and these are the villi shown here, and these are those plica circularis that are shown here. So we have slides that look very similar to this. Do recall that down in the submucosa are a lot of lymphatic lymphocytes and lymphatic structures. So when there's a bunch of them together, we call them the Peyer's patches or Peyer's patches. And they would be there again to protect against any of the stuff in our food out here, that might threaten, because it's in here where we have blood vessels. So this, your small intestine is absolutely adapted for absorption with all this surface area, folds within folds within folds. So here's your plica circularis, these big gentle folds. Then within those are these, right, villi, which look like little fingers. And then on top of each cell that makes up the villas are the microvilli. All right, so that kind of moves you through those. And again, the cells are simple columnar. They are actually called absorptive cells. There are some intestinal pits or crypts, intestinal glands found in here that we'll look at. And then the microvilli are on these, each absorptive cell. There's one shown specifically here, right, this one magnified, and are called the brush border because they appear so, they're so thick they look like bristles on a hairbrush. All right, so in the crevice, the intestinal glands, you would see, right, the enteroendocrine cells. These are the cells that make secretin and cholecystokinin, and there's a number of different ones here. Again, there's a table on page 1116 that outlines all of them if you want to know. Right? There are Paneth cells in here, which are protective. Granular cells, so granules equate chemical warfare. Now, lysozymes, like what you had in saliva, right, that destroy the cell wall of bacteria, will be found here, and cells that would be responsible for phagocytosis. When we looked at the M cells when we were in the lymphatic chapter, right? The submucosa contains duodenal glands in the duodenum, right, which secrete a bicarbonate-rich alkaline mucus. And of course, remember, the duodenum is the first tube after the chyme leaves the stomach. And after it leaves the stomach, it's going to be a very, very low pH. Well, it turns out the small intestine cannot handle low pH, and its enzymes won't work in a low pH, so it's very important that right away in the duodenum we just inundate the chyme with bicarbonate that is produced, of course, by the pancreas, right? And so unique features and purposes here in the small intestine. The mechanical digestion is segmentation and peristalsis, so this kind of shows you this idea of segmentation. There's constriction around this chyme, and then it's isolated and it's pushed back and forth, back and forth, back and forth, back and forth. When it's peristalsis, right, and this is showing you the process through time of mixing it. And what you're doing is you're mixing with all these different secretions from the liver and from the pancreas and from the cells of the intestine itself. So it's, again, it's mixing, pushing the fluid against the mucosa for absorption about 8 to 12 times a minute. When most of the absorption has occurred, then peristalsis takes over. And this is really a propulsive. It moves it very quickly along the tubing, right? In the small intestine, it's about a 3 to 5 hour, and it is intentionally slow to optimize absorption. The reflexes that help control this movement and also prevent you from suffering from blockage we talked about in the first video is that when food enters the stomach, actually, gastrin that is released and promotes all of the gastric phase of gastric digestion, stomach digestion, the digestion by the stomach, I should say. That gastrin also targets other parts. So gastrin targets the small intestine and it targets the ileum specifically. It even targets, of course, the large intestine. So here we have, again, the gastroenteric reflex and the gastroileal reflex. So when food enters the stomach, you really want to be able to tell the other parts of the tube to pick up the pace, it's time to move on, right? So gastroenteric reflex would be the stomach telling the small intestine, hey, increase this propulsive movement because more stuff is coming in. The gastroileal, well, the ileum is the last part of the small intestine, so when this reflex occurs, and it occurs also because when you've eaten, it relaxes the ileocecal sphincter, which is generally very highly constricted, and allows the movement of the food out of the small intestines altogether. All right? Chemical digestion in the small intestine is, there's a lot, right? Because we really haven't done much of anything. It said, as the stomach contents enter and stretch receptors and chemoreceptors stimulate, food is in there, and there's, we've got a situation that we have to deal with. Secretions begin, right? The chyme at this point has partially digested protein, carbohydrates, and very little fat digestion. So we have a lot of bond breaking to do, right? Chyme enters slowly because of that, right, the enterogastric reflex, and it's mixed with these alkaline secretions from the pancreas, and it's mixed with the bile from the liver, and then these other pancreatic juices, right? So pancreatic and intestinal juice will finish. So that's just the term we use for this collective secretions from the pancreas and secretions from the intestines. And if they finish this chemical digestion, then we can absorb. All right? Absorptive cells, right? So intestinal juice is made by these absorptive cells. And these enzymes will complete the final breaking of bonds so that now your proteins, carbohydrates, lipids, nucleic acids are all small enough chemically to enter into the body and into the blood. All right? The large intestines is going to consist of several regions, and most of it identified by the way it's moving. So this is the ileocecal sphincter here to the anus, right? And we're going to finish absorption. Really, it's water and electrolytes. We'll do no chemical digestion. We'll store a lot of bacteria, and the bacteria will help us to make some of our vitamins that we ourselves can't make. We'll form the feces, and we'll eliminate that waste. So off the cecum, you see this right feature, the appendix, which was part of our lymphatic system. And this is a reservoir for bacteria. The colon itself is 4 parts: ascending, transverse, descending, and sigmoidal, because it's S-shaped, and sigma is the Greek letter S. And then the rectum means straight, and then the anal canal with its 2 sphincters will finish up the large intestine. And we'll continue looking at this in the next video.